Talk more about the muscle spindle. Right, so these are the two nerve endings we talked about. The annular spray ending is the nerve ending of the type 1 afferent fiber. The flower spray ending is the nerve ending of the type 2 afferent fiber. And again, as these nerves are afferent, they're taking information about the length of that muscle to the central nervous system, and then the efferent fibers, which are the gamma and alpha, are going to bring that to contract that muscle. Okay, so this just again summarizes what we just talked about. When the muscle is nice and relaxed, right? This is your extrafusal fiber, this is your intrafusal fiber. Right? This is a steady amount of action potential for the fiber, nice steady rate. When the muscle stretches, right, we're going to increase the active potential firing, which is going to result in the contraction of that muscle. Okay, again, this is very important to protect the muscle from injury or overexertion. If you're holding your arms out, someone drops a baby or a book or something heavy into your arms, naturally, you're just going to brace it. Okay? You're not going to brace it and you're still caught on guard, but you're supposed to just naturally contract your muscles to hold that position. Okay? Now, what's important to understand here is, one is that the alpha motor neurons are larger in diameter, so they have a faster conduction velocity. Okay, they have a faster conduction velocity than the gamma motor neurons. So as a result, there's going to be a brief time when that efferent signal is sent from the spinal cord where just the efferent fibers have reacted. Okay? Just the normal contractile fibers on the outside, which are the extrafusal fibers, have contracted. And so the tendon, right, the muscle spindle here, not the tendon, sorry, the muscle spindle inside is going to get wobbly or slack because the outside is contracted, but it is not yet contracted. Why? Because the gamma motor neurons are slower. So they're sort of lagging behind with their contraction. So the extrafusal fibers, innervated by alpha motor neurons, contract first, and then when both of those neurons are on the same page, the entire muscle spindle will then readjust. And it's important that it readjusts so that it can continue to detect any new changes in length. Okay? So having activation of both the alpha and the gamma motor neurons is going to readjust that muscle nicely so that it can continue to detect any new changes or any further changes in the length. Okay? All right, so that was the muscle spindle, and that was detecting or sensitive to changes in length. The second receptor we're going to briefly talk about is the Golgi tendon organ. This one is very simple. Okay, as the name implies, this receptor is located in the tendon of the muscle, right, not in the muscle body. And it's associated with type 1B, again, afferent fibers. So these afferent fibers are in the tendon. They're going to pick up any change in the tension of the muscle. Okay? And if there's too much tension on this tendon, it's going to send a signal to the central nervous system that is going to completely inhibit the contraction of that muscle. And again, this is a protective reflex or protective measure of the muscle. If you're contracting your muscle and the tendon is receiving so much force, it's going to send a message to the spinal cord. You need to stop that muscle from contracting. It's doing the most and it needs to relax, right? So that's going to send an inhibitory signal to the muscle. It's going to stop contracting or slow its contraction rate to protect that tendon from popping off the muscle. Right, and that's the Golgi tendon organ. What we're going to move on to talk about now is the smoothing cardiac muscle. And as we go along, we're going to start comparing them to what we saw with skeletal muscle. Okay, when we go to the cardiovascular system, we're going to talk more in depth about cardiac muscle. So we're going to focus on smooth muscle for a little bit here. So these are the three types of muscle found in your body. We talk at length about skeletal muscle. We talk about its structure, we talk about its function, we talk about its innervation, and that it's voluntary and not involuntary. Okay. Now, smooth muscle and cardiac muscle specifically is similar to smooth muscle in that it is also striated. So skeletal muscle and cardiac muscle together, they are called striated muscle. And that's because they have that striated appearance. So what was the structure that we talked about that gave muscle that stringy or striated appearance? What was the unit of the muscle that had the actin and myosin? What was that called? The sarcomere. The sarcomere. Okay. So the sarcomere being present in both skeletal muscle and cardiac muscle is what gives muscle its striation. Smooth muscle has no sarcomere. Okay, so as a result, it is not striated, and that's why we call it smooth. Okay, no sarcomere meaning no striations, meaning it's just smooth. Okay, it's going to be found in your internal organs, in your blood vessels. So we talked about some of the locations of smooth muscle right when you get the autonomic nervous system, right? Your stomach, your bladder, your uterus has smooth muscle. It's under your involuntary control, right? So it's going to be affected by the autonomic nervous system. So we're not voluntarily controlling our smooth muscle. Now the cells of smooth muscle are different than the fibrous cells of skeletal and cardiac muscle. These are more spindle shaped. They're much smaller and they're sort of spindle shaped. Right? This sort of flat, almost like a squamous cell. A very flat, very elongated, spindle shaped cell. And again, they're much smaller than your skeletal muscle cell, your muscle fiber, or your cardiac muscle cell. They do contain actin and myosin, so although they do not have sarcomeres, they do have actin and myosin. We're going to talk about the arrangement of actin and myosin in a bit. And then they have what's called a dense body. And the dense body is a location or an area on the smooth muscle cell where it connects actin, myosin, and the outer connective tissue. And that's going to be important for transmitting the force of that cell so that the entire muscle can contract as one. So we'll talk about what the dense bodies are in a minute. Now the sliding filament mechanism of contraction still holds with smooth muscle, although there are no sarcomeres. Okay, so this is the smooth muscle cell. Another thing that we want to point out here is that they are mononuclear, so they only have one nucleus, as opposed to multinuclear skeletal muscle cells. So they have one nucleus, much smaller, rounded shaped cell, and this is the orientation of actin and myosin. So the thick filaments are these yellow little squares here. The thin filaments are those fiber strands, and they are running more obliquely or diagonally, as opposed to the parallel organization of the skeletal muscle fiber. Okay. Now, also important to note here is that the entire myosin head, right? So this is the myosin head, this is the actin, and the entire myosin head is going to move along the actin filament, okay? And this is going to cause a longer contraction of the smooth muscle, all right? So having longer actin, longer myosin, like having a diagonal arrangement, and having the entire myosin head move along the actin filament, or move along the thick filament, is going to cause a slower, longer contraction in smooth muscle. So this is what a smooth muscle cell looks like relaxed. This is what a smooth muscle cell looks like when it's contracted. Okay, very different from skeletal muscle. Okay, so now we're going to talk about the steps of smooth muscle contraction. Okay, and they are quite different from skeletal muscle contraction. Okay, so we have our smooth muscle cell, right? It's sort of spindle shaped here. So this is a smooth muscle cell. Now, on the membrane, of this muscle cell is going to be a voltage-gated calcium channel. Okay, we're going to talk about why that's important in a minute. Inside the muscle cell, we have the endoplasmic reticulum, and this is where calcium is being stored. Okay, so this is the endoplasmic reticulum. And this is important for the storage of calcium. Okay, so let's just draw some calcium floating around in here. All right, and there's also lots of calcium in the extracellular fluid. So this fluid, or the space around the outside of the cell, has lots of calcium, as well as the storage source, which is the endoplasmic reticulum. So the first step of smooth muscle contraction, or step 1 here, right, is that we're going to depolarize this membrane. So we depolarize the membrane, which causes calcium to influx. All right, so calcium is going to move into this voltage-gated calcium channel as a result of the change in the membrane potential, or change in the voltage of that membrane. Now, when the calcium rushes into the cell, that is going to trigger step 2. Okay, that's going to trigger step 2 here. And having lots of calcium in the cell, inside the cell, is going to trigger more calcium to move from the endoplasmic reticulum into the cell as well. Okay, so calcium is also moving from the storage area into the cell as well. And this phenomenon is called calcium-induced. Right, this is step 2, calcium-induced calcium release. And all that means is the presence of calcium in the cell sort of triggers a positive feedback response so that more calcium rushes from the sarcoplasmic reticulum into the cell, or into the cytoplasm. All right, that's step 3. Step 2. Step 3, right, is that calcium is going to bind to a molecule in the cell. So there's a molecule in the cell called calmodulin. This is called calmodulin. Right? Calmodulin. And when calcium binds to this calmodulin, we put a big old calcium here. This forms a complex called the calcium calmodulin complex. Okay, the calcium calmodulin complex. All right? And now that complex is going to do something very important. It's going to trigger step 4. So step 4 is that calcium and calmodulin bound together in this complex are going to turn on this enzyme called MLCK. This out here. So we got MLCKs, this enzyme here. And when this interacts with this, it's going to activate it. Let's put a little plus sign here. So the activation of MLCK is going to, so it's, let's write out what this actually is. So this is called myosin light. Myosin light chain kinase. And this is a fancy way for saying that this enzyme is going to phosphorylate or add a phosphate group, right, or energize the myosin head. Okay, so this enzyme is basically going to energize or activate the myosin head by phosphorylating it, by adding a phosphate group. Okay, remember it's a kinase. So let's write that here. So this is going to phosphorylate myosin head. And doing so is going to activate cross-bridge cycling. Which is going to lead to contraction, right? Contraction. Okay. Now, when the muscle needs to relax, okay, it's not going to be as easy as it was in skeletal muscle, right? When the myosin is not just going to float away or not be activated. We're going to need another enzyme here, another enzyme to undo this process. Okay, and this is called a phosphatase. And a phosphatase, like as the name implies, is going to grab the phosphate group off of those myosin heads. But in order to be activated, it needs a trigger. And the trigger to activate phosphatase is the decrease in calcium. So I'll put here decrease in calcium activates phosphatase to unphosphorylate myosin heads. And this is where we have no contraction. Okay? So that phosphatase is basically going to compete with MLCK, depending on whether or not there's calcium. So if there's calcium in the cell, all right, that phosphatase is not going to be active. MLCK is going to be dominated. All right? When calcium starts to leave the cell, right, due to this calcium ATPase pump, because those are also present here, we'll talk about those in a minute. When calcium starts to leave the cell, then the phosphatases are activated, and they're going to go around and help grab the phosphate groups off of the myosin head. Okay?